An ECG is a recording of cardiac action potential throughout the cardiac cycle. These action potentials are recorded in millivolts, and are represented in measurable deviations from the isometric line. This isometric line represents the cardiomyocyte's resting membrane potential, and deviations represent the stages of action potentials within the cardiac conducting cells. To properly understand what an ECG is and what it represents, we must first understand the mechanism of action potentials. An action potential can be considered as the electrical stimulus of living cells. Each action potential occurs through a number of steps to complete a full cycle. However, each action potential is comprised of two main steps: depolarization and repolarization. Depolarization is the process of the intracellular space becoming more positive, resulting in an increase in voltage, while repolarization is the process of the ion gradient returning to the resting membrane potential. The resting membrane potential of most cells is between -50 and -70 mV, with the intracellular space being more negatively charged, and the extracellular space being more positively charged. However, in cardiac cells, this resting potential sits at around -90 mV. This resting potential is established as a part of an electrochemical gradient, with a greater ratio of sodium ions in the extracellular space, and a greater ratio of potassium ions in the intracellular space. As these ions shift across the cell membrane through specific ion channels, action potentials occur. Depolarization occurs as the intracellular space has a rapid influx of sodium ions across the cell membrane. These ions cross the cell membrane through voltage-gated sodium channels that open temporarily, to allow this depolarization stage. These sodium-gated channels open in response to a slight voltage increase from -90 to -70 mV as the action potential continues along the neighboring cell. This influx of sodium increases the voltage of the intracellular space, until the intracellular voltage reaches around +50 mV. Once this voltage threshold is achieved, the voltage-gated sodium channels shut. Inversely, repolarization occurs after these voltage-gated sodium channels shut, and potassium-gated channels open, allowing potassium to flood out of the cell, and the electrochemical gradient returns to the resting membrane potential. A cardiac action potential occurs across 5 key stages involving movement of these ions across the cell membrane. These stages are 4, 0, 1, 2, 3, and then back to 4. Stage 4 is the state of the resting membrane potential. At rest, most neurons are semipermeable to sodium, potassium and calcium, meaning that small amounts of these ions can move across the membrane. However, cells are most permeable to potassium, and this has the greatest influence on the resting membrane potential. The initiating phase, also known as stage 0, involves the depolarization of the cardiomyocyte. This is stimulated by a voltage increase from action potentials that occur in the adjacent cells through gap junctions. As the voltage in a previous myocyte along the conduction pathway increases, the threshold of 70 mV is reached, and the sodium-gated ion channels open, allowing sodium to flood through the cell membrane, and increase the intracellular voltage to +50 mV. Stage 1 begins as the sodium channels rapidly shut once the voltage threshold is reached. During this initial phase, potassium channels open and shut rapidly to allow a partial exchange of potassium ions, but the majority of potassium ions remain in the cell. Stage 2 is also known as the plateau phase, due to the membrane potential remaining almost constant, as the membrane slowly begins to repolarize. During this phase, delayed potassium channels allow some potassium to leave the cell, while calcium channels allow the movement of calcium ions into the cell. These calcium ions are responsible for the contraction of the myocardium. Across stages 1 and 2, the absolute refractory period also occurs. This period is where the sodium channels remain shut, and cannot open regardless of the degree of stimulation that may occur. Stage 3 is the rapid repolarization phase where voltage-gated potassium channels open, and potassium floods out of the cell to restore the membrane potential to negative 90 mV. During this stage, the relative refractory period also occurs. This period is where the sodium channels remain shut, but can be reopened, however a greater than normal stimulus is required for this to occur. Once the resting membrane potential is achieved, stage 4 recommences. All of these stages are passive transport methods, meaning that they require no energy to occur. They simply require ion concentration gradients for passive diffusion across a selectively permeable membrane. At the completion of an action potential cycle, the ion concentration gradients must be returned to normal, with the sodium being moved back to the extracellular space, and the potassium to the intracellular space. These concentration gradients are maintained by the action of sodium-potassium pumps located throughout the cell membrane, which are an active transport method that allows the membrane potential to be maintained. This active means of transport requires the presence and binding of adenosine triphosphate, known as ATP. As ATP binds to the sodium-potassium pump, the electron potential from one phosphate group results in activation of the pump, leaving adenosine diphosphate, or ADP, as a byproduct to be recycled. When this occurs, 3 sodium ions are actively pumped out of the cell, and 2 potassium ions are pumped into the cell. This continues throughout the cardiac cycle to restore the concentration gradients of sodium and potassium for the next action potential. These action potentials occur throughout the myocardial cells along conduction pathways. In a healthy person, cardiac action potentials are self-initiated within the sinoatrial node, located in the right atrium. As the voltage within these cells increase with ion transfer during depolarization, there is a slight voltage increase in the adjacent cells through gap junctions. This slight voltage increase stimulates the depolarization of the next cell in a domino effect. From the sinoatrial node, the action potential travels from the right atrium to the left atrium through the Bachmann's bundle, and to the ventricles via the AV node. The AV node causes a momentary conduction delay, allowing the artery to empty before initiating the ventricular action potential. From the AV node, the action potential travels through the bundle of His, then branches out to the left and right bundle branches, and finally continues to the myocytes through the Purkinje fibers. This conduction through action potentials is important to understand because this is what is reflected in an ECG. A point of note is that although the voltage potential graph reflects the same events, they differ slightly in their exact representation and are not to be confused. The voltage graph demonstrates the polarity of a single cell which becomes either more positively, or more negatively charged. An ECG on the other hand represents both positive and negative voltage shifts along the cardiac cells either moving towards, or away from specific leads. Each individual lead on an ECG recording is slightly different in shape. This is due to each lead recording the electrical activity from different directions. When the electrical activity of the heart travels towards a lead, whether depolarization or repolarization, it results in a positive deflection. When the electrical activity travels away from a lead, it results in a negative deflection. This is important to note, as a downwards deflection does not represent a negative charge like it does on a voltage graph, but rather a positive or negative shift traveling away from that lead. Deflections from the isometric line in a normal ECG are represented as P, Q, R, S and T. The P wave represents atrial depolarization. The QRS complex represents ventricular depolarization, and the T wave represents ventricular repolarization. While atrial repolarization does occur, it is not seen on a normal ECG due to being hidden by the QRS complex, which is of greater magnitude. This is called sinus rhythm, and deviations from this are identified as cardiac disorders, or dysrhythmias. The height of the deflection also represents the amount of electricity flowing in that direction. The lead with the most positive deflection is closest to the direction the heart's electricity is flowing. If the R wave is greater than the S wave, it suggests depolarization is moving towards that lead. If the S wave is greater than the R waves, it suggests depolarization is moving away from that lead. If the R and S waves are of equal size, it means depolarization is traveling at exactly 90 degrees to that lead. This is also important to understand, as it is not only the size of the action potential, but also the distance of the lead from the action potential that dictates the height of the deflection. This is why the limb leads on an ECG have the smallest complexes, and the V leads have the largest.